I so appreciate that you have taken the opportunity to listen to this presentation titled The Wisdom of Your Cells. For in this science that we're going to hear is a new biology, a biology that will profoundly change civilization and the world that we live in. This new biology takes us from the belief that we are victims of our genes, that we are a biochemical machine, that life is out of our control, into another reality, a reality where our thoughts, our beliefs, our mind totally control our genes, our behavior, and the life that we experience. The interesting part about this biology is that it's really based on current modern science, but with some new perceptions added in that are relatively recent. And the significance of all this is that since we didn't apparently pick our genes, and since we can't change our genes, and since the belief is that genes control who we are, then we have to look at ourselves as recipients of programs, and that we must then try to live out our life given the programs that we received at conception. However, there's a new understanding coming, and this new understanding is at the leading edge of science, and I'm trying to bring this down to the population for the reason is that this new science takes us from victims to creators. And the relevance about that is that we are actually very, very powerful in creating and unfolding the lives that we lead. So that what I would like to provide you with is a scientific foundation to give you an understanding of who we really are. This is actually knowledge of self. And if we go back and understand the old axiom, knowledge is power, then what we're really beginning to understand is knowledge of self is knowledge of self-power. And this is what I think that we will be able to get from understanding the new biology. My first introduction into biology was in second grade. At that time, I remember I went to school early in the fall that year, and when we were all getting ready to settle down in the classroom, Mr. Bangelsdorf brought in a microscope, and he was going to show us the cells. And I remember how exciting it was that we were all lined up as he focused the image of the microscope on some amoebas and paramecia in this little field of swamp water. And all the kids were getting up there, getting a chance to look at it. And finally, I got my chance to look down the barrel of the microscope, and there I saw the first time these little cells moving around in their own environment. And in that 7-year-old mind of mine, I looked at these and realized that these cells were not just bumping around and looking like little pinball machines where they were just bouncing off the walls, that these cells looked very busy. They were industrious. They were looking around at things. They were checking things over here, and then the cell would move and check things over there. Immediately at that stage, I began to realize that these were actually sentient beings. So even in that young mind, I started to make a connection to realize that there's these life forms down there, and that in consideration of my own particular life and the background I was coming from and the troubles of function and dysfunction as most of us experience in our family growth, when I saw this other world, it offered me another place to go and see life from like looking over a cell's shoulder. Well, it was interesting because I didn't realize that great introduction that I had back in second grade was actually going to shape the rest of my life. About 12 years later, I ended up in graduate school at the University of Wisconsin, where I graduated from conventional microscopes into electron microscopy, and then had a further opportunity to look into the lives of these cells. And the relevance about that is the lessons that I learned from these cells profoundly changed my life and gave me insights about the world that we live in that I would like to share with you. So this is more or less a story from our cells. In the opportunity of using electron microscopy to look at cells, not only did I see the cells from the outside, but I was able to fly through their interior and go through the cell's anatomy and understand the nature of its organization, its structures, and its functions. And this was just a wonderful opportunity to go visit this other world. I used to actually laugh that they were actually paying me for this job, where I get to go sit in this room with, it actually looked like Star Trek, with this giant electron microscope, a column of steel and pumping machines and LEDs and lights flashing, and then get a chance to fly into cells. So as much as people talk about flying into outer space, I was going into inner space and seeing vistas that no people have ever seen before. So it was this great opportunity, and that through this understanding, I started to get a greater appreciation of the nature of life and the nature of cells and our involvement with our own cells. It was at this time that I also started to do some training in cell culturing. And in fact, in about 1968, I started cloning stem cells. Well, today stem cells are a hot topic and everyone thinks, well, wow, where did they come from? Stem cells, just a couple of years ago, nobody talked about them. Essentially, I had been working on stem cells for about 40 years. During that time, I was really given the opportunity to create my own culture systems and then observe and track the fate of cells and then try to look at their structure and their biochemistry as they change their life, as they change their fate, to get an idea of the mechanisms that were controlling cell biology. So in 1968, I was doing my first cloning experiments, and it was under the guidance of Dr. Irv Koenigsberg. Now, Irv was a brilliant scientist, and he was the man who actually created the first stem cell cultures. Well, during this process, on the very first day of setting up cultures, I was given some advice by Irv that I didn't realize until 20 years later how important this information was to the understanding of life on this planet. Essentially, I had just completed the cell culture process, inoculated the cells into a petri dish, and was about to put the petri dish into the incubator, and Irv stopped me and he said a very important guidance about the work we were doing, and he said this. He said, when you come back in tomorrow, Bruce, and you look at these cells, if the cells don't look right, if they look unhealthy, before you really try to alter the cells, the first thing you have to look at are the conditions of the culture environment. So basically what Irv was really leading me on to was an understanding about the influence of the environment and the expression of health or dis-ease among biological organisms. Throughout the rest of my research, I started working with these cells, these stem cells, and specifically the stem cells I was working with were called myoblasts. Myo means muscle, blast means progenitor. So these are stem cells that are not differentiated. They don't express any particular fate, but you can identify them because of their very unique spindle shape. What I was doing in my cultures was isolating these spindle-shaped cells and then allowing them to replicate and form a large colony so that all the cells in the colony were genetically identical, derived from the same precursor cell. So I was working with a uniform, genetically identical population of cells. Through my research, I started to realize that when I put my cells in the culture dishes with the conditions that support muscle growth, of course, the muscle cells evolved from these undifferentiated cells, and I would actually end up with cultures with giant contractile muscles derived from these single-cell precursors. However, during the course of my experiments, I started to realize that if I started to change the environmental situations and the components of the culture medium, that the fate of the cells would be altered. So that I would start off with my same myoblasts, my muscle precursors, but in an altered environment, let's say environment B. When I put the cells into those petri dishes, they'd actually start to form bone cells. If I further altered and manipulated the culture conditions, and let's say create another condition called environment C, and put the same muscle precursors in that particular dish, those cells started to become adipose or fat cells. The results of these experiments were very exciting because what it really revealed was, while every one of the cells was genetically identical, the fate of the cells was not controlled by the cells, but was controlled by the environment in which I placed the cells. And why this was really relevant is because at the time I started doing these experiments, I also started teaching students, undergraduate students, and later teaching medical students at the University of Wisconsin, the conventional understanding that the fate of cells was controlled by genes. And yet in these experiments, it was clearly revealed that the fate of cells was more or less controlled by the environment that I was putting them in. So what I started to go into was this other view of what was controlling life. Well, my colleagues, of course, were very upset with my work because everyone was then on the bandwagon for the Human Genome Project that was getting tooled up, and everyone was really so far in support of the genes control life story that when my story revealed how the environment would alter the cells, when they heard that story, it sort of made them a little uneasy, and then they talked about it as an exception to the rule, which to me was a little bit of a put down because I was thinking, well, there are no exceptions to the rule. There's some reason as to how the environment is controlling these particular cells. So I ran into a conflict because I realized that I was teaching medical students the conventional story of DNA and genes controlling life, and yet the cells in my laboratory were revealing a completely different understanding of life where all the cells had the same genes, but their fate was really determined by the environment. Now I have a completely new understanding of life because as I started to follow this research, it led to a new way about teaching people about cells. So let me start with this first understanding that I was contributing to my medical students about the nature of cells and cell biology. Number one, when we look at ourselves as a single individual human, so when I look in the mirror, I see Bruce. When you look in the mirror, you see yourself as a single person. Well, it's an interesting observation, but it's a misperception, meaning this. If you really understand the nature of who you are, you realize that you are actually a community of about 50 trillion living cells. Each cell is a living individual sentient being. Every cell has its own life. Every cell has its own functions. Every cell interacts with other cells in the nature of a community. So while we stand and look at ourselves in the mirror as a singular entity, if I would reduce you to the size of a cell and dropped you inside your own body, you wouldn't recognize yourself as a single entity. You'd see a very, very busy metropolis of trillions of individuals living in harmony, especially if we're in health, under one skin. So number one, we are not really a single entity. We are collective consciousness of upwards to 50 trillion individual amoeba-like cells, so that we actually represent a community. This becomes really relevant when we understand that health is when there's harmony in the community, and dis-ease is when there's a disharmony that tends to fracture the community relationships among ourselves. So number one, we are a community. Fact number two, well, we look at the human and think about the human as being this magnificent biological organism with all the functions that we have built into our body. Here's an interesting fact. There's not one new function in the human body that's not already present in every single cell. So, for example, well, you have a digestive, respiratory, excretory system, a musculoskeletal system, an endocrine system, a reproductive system, of course, a nervous system, and even immune system. Every one of those functions exists in every one of your cells. So every cell has all of the functions that a human can express. Interesting point, of course, is as humans made out of cells, just the logic says, how can we create something that our cells can't manufacture or our cells can't do? We are the reflection of our cells. So when we start to look at biology, we can look at a human and look at all of its functions, or at a more simple level, we could go down and look at a cell and look at the same functions present in a cell. And what's very interesting about this is that the pattern and structure and behavior of a cell is replicated in the human. In fact, we are made in the image of a cell. And this is very helpful for biologists because we can do research on cells and then apply that information to understanding the nature of the human. Well, this is going to be a very important point through the rest of the discourse on this story on the biology of belief. For this reason is that while we'll be talking about cells, we must remember that we can interchange a human or a cell in regard to the basic behaviors and needs. Between the two, they're exactly the same. So in conclusion, in this regard, when I started teaching the students about cell biology, I got them to understand, A, that they were communities, and B, that the cells that make them up can do all of the functions and express all of the behaviors that a human has, and therefore we can really talk about them as being one and the same in this understanding. So it's really wonderful then to get into the nature of talking about cells, because what we're going to see is the character and behaviors of our life are reflected in these very simple structures. So I'm teaching in the medical school, and I'm teaching the fundamental nature of what is called the medical model. And the medical model is the perception that the human biology represents a biological machine comprised of biochemicals and controlled by genes. So that when a patient comes in to see a doctor and there's something wrong, the tendency in the belief system of the student educated with this belief is that the patient has something wrong with their biochemistry or genes, which can be adjusted and can lead them to health. When I started to confront my colleagues and say, well, I'm teaching this genetic story, I'm still having trouble explaining how the environment is controlling my genes, they had a tendency to disregard this information because it didn't fit the medical model. So at some point, I started to realize that I had to leave the university because I found great conflict in teaching the students what were apparently facts about what controls a cell, and yet getting completely different understandings of control from the cells in my culture. During that opportunity, when I was outside the university, I had a chance to read into physics, which we're going to talk about later on as well. And the reason why this is important is because this is again where I found the information that I started to read in the physics did not conform to the science that I was teaching in medical school. What is physics related to medicine? Well, simply this. Physics, by its definition, is the science of the mechanisms of the universe. In fact, physics and mechanics are synonyms with each other. That's why you can say quantum physics or quantum mechanics, and they represent exactly the same thing, or Newtonian physics and Newtonian mechanics. Well, the relevance was that as I started to look into the world of new physics, the world of quantum physics, I started to realize that the mechanisms that are described in the nature of that physical view of the universe completely collided with the mechanisms that we were teaching in medical school, which were based on Newtonian physics. So we're going to talk about the nature of the control mechanism. We're going to talk about the fact that it's not the genes, but we're also going to bring in the concept of a new physics, which was not introduced in medical school and currently is still not introduced in medical school, and that is the relationship of quantum physics to the operation and function of cells. And this becomes an important turning point for us because we're going to leave behind some very old beliefs that we bought as fundamental truths and enter into a new understanding of biology by adjusting the biology in regard to the new understanding of control and the new understanding of physics. The question is, how come it's so hard to get people to perceive new insights into the science of human biology, the medical model? And the answer is because we bought a belief, and we've invested so much of our time and so much of our history into this belief that when changes start to become needed in the understanding, it's very difficult to introduce them because of the established point of view that has been concretized in our knowledge. And the issue is, so how come our belief in the medical model is so strong? Well, the answer is we must first go back to the origin of modern science. So we start to go back into the late 1500s and 1600s to see what kind of foundational beliefs were introduced when science began to introduce the world to a new way of seeing how life and the universe was working. Before conventional science, of course, there was a science, but it was a science delivered by the church. It was a science called natural theology, in which science was infused with the spiritual domain, and that God's hand was directly involved in the unfoldment and maintenance of the world that we live in. So natural theology was really a belief that God's image was expressed through the nature that we lived in. And actually, the early form of science at that time, natural theology, had a mission statement, which is really to understand the nature of the environment so that we could learn to live in harmony with that. So it was basically learning how to live in harmony with God, considering that nature and God were so well connected. However, through the abuses of the church and their insistence on absolute knowledge and the efforts of suppressing new knowledge, there came a point where we experienced what's called the Reformation. And the Reformation, which was really precipitated by Martin Luther, was a challenge to the church's authority. And when that challenge was successful, it opened the doorway for science, because before then, when science would start to study the realm of nature and start to find information that didn't agree with what was being taught in the religious schools, these scientists were actually forced to recant their beliefs or undergo the so-called kangaroo trials of the Inquisition, which forced them to relent and go back and accept the information that was being taught by the church and let go of their views of visions that they acquired through their own scientific efforts. Again, this was to keep the nature of absolute knowledge in the hands of the church. But as I said, after the Reformation, when there was an opportunity to question the beliefs about the universe, this is when science was grounded and actually became what was called modern science. The foundational research that shapes modern science was provided by Isaac Newton. Isaac Newton is the physicist who primary studies were involved understanding the nature of gravity and the movement of the planets in the solar system and the moon around the Earth, etc. And to understand the nature of how these movements occurred, Isaac Newton created a new mathematics called differential calculus and then tried to put data in about the objects in the solar system to create an equation where he could predict the movements of the solar system. The reason why the prediction is necessary is because modern science said, unlike the past where religious beliefs were just based on religious beliefs, science wanted to know if there were truths that could be understood. And so science introduced the concept of an ability to analyze the environment and understand the mechanisms that provide for the environment. But in that scientific analysis, it involved recording of data, the setting up of experiments based on hypotheses, and then recording that data from the experiments to see if the hypotheses and the actual observations fit. The conclusion is simply this: science identified truths as things that were predictable. So what was Isaac Newton doing? He was creating equations where he could predict the movement of the planets in the solar system, and by doing so, if he could predict it, then his science would reveal a truth about the nature of the universe. In his equation, Isaac Newton included important things such as the mass of a planet, the acceleration of a planet, the vector in which direction that planet was trying to move, and he put these elements into his equations and was successful enough that he was able to actually predict the movement of the planets in our solar system. What was really, really relevant about Newton's work was this. In his equation, he only put material characteristics of the universe, the physical mass of, let's say, a planet, the acceleration of this mass, gravity, which was a factor of the mass. The more mass something has, the more gravity it has, and basically created these equations. But here was the exciting part. In his equations, Newton didn't put God, he didn't put spirit, he didn't put invisible forces, he just put material characteristics. And what was then relevant with the results of his study is he said you can understand the nature of the universe by only studying the material components, that it's not necessary to invoke God or spirit or invisible forces in how the universe works. This is the foundational understanding that seeded what is called Newtonian physics. Newtonian physics characteristic is it perceives of the universe as a machine made out of matter, and says if you can understand the nature of the matter that comprises that machine, then you will understand the nature of nature itself. So all of a sudden, the science that was evolving, called modern science, left behind the concept of invisible forces and spirit, and said, let's just focus on matter, that everything in the universe could be understood by looking at its material composition, and by studying that, you will understand the nature of life. So the new field of science took a whole different tack than where we were coming from, where we looked at our fate being controlled by spirits and forces beyond us. The new science says that we're controlled by the material character of which we're made. This also introduced a new mission statement of science. Since it is now perceived that the universe was a machine made out of matter, it also suggested that if we understood the nature of the machine, we can control and dominate the machine. So the mission of science was actually to control and dominate nature, which is completely different than the former mission of science under natural theology. That mission of science was to learn to live in harmony with nature. Now with modern science, we were out to control nature. Well, the issue of control in regard to biology becomes a very important point, and that is, well, what is it that controls us? What is it that controls the traits that we express? Well, there wasn't really any idea where to go with that until Charles Darwin created his theory of evolution on the origin of species by means of natural selection. In Charles Darwin's work, he offered a piece of information that most everybody of the time was quite familiar with, and that was this. The traits of an individual are connected to the traits of the parents. Back in those days, there was a lot of animal husbandry and plant breeding, and people were just breeding all kinds of animals as pets and as farm animals, and it was very clearly established that the traits of the parents would be expressed to some degree in the offspring. Well, if we go back into understanding what Newton said that the universe is made out of matter. To understand the universe, you can just look at the matter and don't have to invoke invisible forces. Since life forms are parts of the universe, then according to Newtonian physics, life forms represent machines made out of matter. And if you want to understand anything about those machines, then you could take them apart, a process called reductionism, and then upon taking them apart, study the individual pieces of matter, and then see how they work, and then when you put all the pieces back together again, you'd have an understanding of the whole. So now Darwin comes in and says, look, the traits that an individual expresses are connected to their parents. When you apply Newtonian physics, it says that matter is the only thing that we should be focusing on. Then all of a sudden it became obvious that something in the sperm and egg that come together during the process of fertilization and result in the formation of a new individual, something physical in those germ cells, the sperm and egg, must be carrying the traits, or something that controls the traits in the offspring that will be derived from those germ cells. So the nature of science got immediately myopically focused on saying, what is it? What are those physical things inside a cell that control life? Well, in the early part of this work, it wasn't really clear what those control elements were. Now, Darwin didn't understand the nature of genes, so he actually talked about things called gemmules, which were hypothesized little pieces of matter that controlled the traits, and so that there was like gemmules that make the arm, and gemmules that make legs, and gemmules that make the stomach, etc., that each part of the body had these gemmules, and that in the germ cell there were going to be a collection of these gemmules that will be passed on to the offspring, and they will control the structure and function of the parts of the offspring. So science started to try to identify what could these gemmules be. And really what this led to was studies of dividing cells that were first studied around the early 1900s and looking at cells as they divide through microscopes that were getting more sophisticated at the time. They began to see these string-like structures that were present in cells that were beginning to divide. These string-like structures were called chromosomes. They started to recognize that the chromosomes were connected to the heredity. So all of a sudden the focus said, okay, we're beginning to find what controls life. In the germ cells are pieces of matter, and these pieces of matter are called chromosomes, and are connected to the transference of traits. Well, the interesting thing about the chromosomes when they started to isolate them and take them apart is that chromosomes were made out of 50% protein and 50% DNA. And they tried to say, okay, which is the part that controls the traits? Is it the protein or the DNA? Because their effort was to take this thing apart and get down to the very nuts and bolts of life, and they were trying to identify which of these components control the traits. Interestingly enough, while the chromosomes were identified around 1900, it was only in 1944 that we actually identified which of those components carried the genetic traits. And it turns out that the experiment by Avery, McLeod, and McCarty in 1944 provided us with the insight through this very simple observation. Avery, McCarty, and McLeod were working with two kinds of bacteria, one type R and one type S. So they're growing the type R bacteria in cultures, and these bacterial cultures, when they grow, make little tiny dot-like colonies, like little BBs all over the Petri plate. The other species of bacteria they're working with, type S, the colonies form large, irregular-shaped puddles, so that they had two different traits. The type R had the BB shape. The type S had the big puddle-shaped colonies that were produced when the cells grew on agar. So the question is, can you change the trait of the colony from one species by using the chromosome material from the other species? Well, the chromosome of type S, they isolated this one, and the type S makes the big puddle-like colony, and they separated the chromosomes into two components, the protein and the DNA. And then they started growing cultures of type R that make the little BB-shaped cultures. And what they did is, in one set of cultures, they added the protein from type S, and in another set of cultures, they added the DNA from type S, and then let them grow. What they saw was amazing, was that the type R cultures, little BB-shaped normal cultures, when incubated with DNA from a different species, the type R changed its morphology, changed its shape, and actually started to express the big puddle-like colonies of the type S bacterium. And so what they found was, while the chromosome was made out of protein and DNA, apparently it was the DNA that contained the trait-controlling material, because by adding the DNA from one species to another species, they altered the traits of the recipient species. So all of a sudden the world got very excited because they said, oh my goodness, after all these years we finally have gotten down to identifying what is it? What is that genetically controlling material? It appears to be the DNA. So the research didn't take very far from 1944 to 1953. In 1953, the world was stunned by the results of work by James Watson and Francis Crick. Watson and Crick published an article in Nature revealing the structure of the DNA molecule. It showed that this molecule was a double helix, and that each helix was actually a linear chain like beads composed of 4 different kinds of subunits called the DNA bases. Many of us are familiar with them because the 4 letters A, T, C, and G are frequently used in describing the nature of the genetic code. Adenine, thymine, cytosine, and guanine. And what Watson and Crick revealed was that each strand of the double helix contained a sequence of these bases, and that each of the genes, which are sequences of these bases, had a unique pattern of bases, that the bases actually were blueprints to make the proteins of the body. So the DNA were linear blueprints to make the protein building blocks. So now we must stop for a second and say, well, what are the proteins? Well, the proteins are very large molecules called macromolecules. There are over 100,000 different kinds of macromolecule proteins that are building blocks to make a human body. So that says there are 100,000 different kinds of proteins. Each protein also has a unique structure, but it turns out that the structure of the protein is encoded in the blueprint of the DNA, so that the DNA strands were linear blueprints that would then code for the production of the proteins. So we had over 100,000 different proteins, and the genes were the DNA segments that were the blueprints for each of these proteins. So what Watson and Crick revealed was the nature of how the code was put into the blueprint. It was the sequencing of those 4 bases, A, T, C, and G, so that all of a sudden the genetic code was broken. I remember back in 1953, being relatively younger then, the headline that came out in the New York paper was a tabloid, and the entire front page in giant block letters was "Secret of Life Discovered." And from that point on, biology has been so wrapped up in the genes, because after 100 years from Charles Darwin's suggestion that heredity was carried by substance in the germ cells, we finally identify what that substance is and know the code. And science was really excited because, remember, science's mission statement was to dominate and control nature. With the finding of an understanding of the genetic code, scientists saw that, the future, the mission that they've been working on was really near, that by understanding the nature of the genetic code, that we could change the characters of organisms, change the traits, and therefore we would succeed in the mission of what? Dominating and controlling nature. So there was a big headlong rush into the Human Genome Project to try to understand the nature of the genes. And the relevance about all this is that when we started to step back and say, well, what did we find? We found that the traits of an organism are controlled by the genes that are transmitted from parent to child, and the genes were more or less the fate of an individual, so that conventional belief was that at the moment of fertilization, you get a set of genes from your mother, you get a set of genes from your father, the two come together, and then from that set of genes, it presents a program or fate map of the future of that individual. At first we thought these genes only control just the physical form. But the more they started to manipulate the genes, they also started to see that there were influences on behavior and influences on emotion. So all of a sudden the genes take on more profound meaning in our new world of biology, because all of a sudden it describes all the characters and traits of a human are apparently controlled by these genes. Well, what's interesting was that science didn't really stop at that point. Francis Crick and James Watson said, wait a minute, the DNA is the blueprint for the protein. The proteins are the physical elements that give us our behavior and give us our structure, so that the DNA is pre-coding our behavior and our structure. And yet the one last question was, well, if DNA controls the proteins and the proteins control life, one last question is, what is it that controls the DNA? Because that would be going up to the last rung of the ladder to find out what is ultimately in control. Well, they did an experiment on the double helix. Remember I said there were two helical chains of DNA enwrapped around each other. That the DNA helix, each chain is a sequence of A, T, C's, and G bases that have a unique sequence for each gene. And what they found is that in the two chains that make up the helix, one chain is a physical complement of the other chain, so that they're not two random chains of DNA. In chain A, the bases that are lining from one end to the other of the chain are complemented by bases on chain B, the other piece of the DNA. What this really means is that one side of the DNA helix may have the linear code for a protein, might be the blueprint for the protein. The other side is a physical complement. Now, what I mean by physical complement is simply this. The chains are made out of these bases, which are A, T, C, and G, adenine, thymine, cytosine, and guanine. But what it turns out is that an adenine will plug into a thymine, a guanine will plug into a cytosine. And the same thing, the TA, thymine and adenine, will always complement each other, and the GC pairs will always complement each other. So on chain A, let's just say I have the following base sequences, A, T, C, and G. The complement strand, the other strand of DNA, will have the complete complement. So the complement of A is T, the complement of T is A, and so basically what I start to find is that, well, I have A, T, C, and G on one side, I have T, A, G, and C on the other side. Basically what it says is that the two strands are mirror images of each other. Well, it might sound complicated, just understand this, that they are complements. So here was the last experiment that really shaped modern biology. They split up a DNA helix into A strands and B strands and separate them, so they only have a single strand. And let's say they take the A strand and they stick it into a beaker with all the building blocks of DNA, all those A, T, C, and G subunits. When they put the A strand and just soak it in that beaker with all the subunits and leave it for a short time, and then pull that A strand out, what they find is that the A strand will actually serve as a template to code the B strand, and so what I put in as a single strand, when I pull it back out, is a double-stranded DNA again, and that the second strand is a physical complement of the first strand. So that what this really revealed was DNA was responsible for copying itself. So if you take a DNA molecule and split it up, each of the two chains will then form a complementary chain and repair themselves so that you end up with two double helices. So that in a cell, a chromosome will have two double helical strands. The chromosome will split into two, and then each of the strands will make a complement of itself, and then you end up with two chromosomes. One of them will go into one daughter cell, one will go into the other daughter cell. The conclusion of this experiment was basically very fundamental and very important in shaping the biology we have, because it said this. DNA controls the protein, and the protein represents our bodies. And DNA controls itself, meaning that DNA is responsible for replicating its own self. So immediately what they said is, oh my goodness, that's the whole hierarchy of information. DNA controls itself, and DNA controls the protein, and that led Francis Crick to describe what is called the central dogma. And this is what I was teaching in medical school. The central dogma is this, that the flow of information in biology goes from DNA to RNA, and RNA is like a Xerox copy of the DNA, and that the RNA is used as the physical template to make the protein. So basically, the unfoldment of information is DNA goes to RNA, goes to protein. We, our physical bodies and our behaviors, reflect the protein part. But then when people say, well, where did you get your physical traits? Where did you get your behavioral traits that are manifest in the proteins? The answer is you go back up the ladder and you say, well, the proteins were coded from the RNA, but the RNA is a copy of the DNA. And so basically it says that life is controlled by DNA. That is the central dogma. And the relevance about that is that once we start to believe that, then we believe also in what is called the primacy of DNA, which is another way of saying the same thing in textbooks. Primacy meaning first cause. So when they say, well, tell me about that trait or character that you express in your life. And then a scientist would have to say, well, the first cause, the primary reason, is the DNA, because the DNA is responsible for giving us the pattern and shape of the proteins, which in turn give us the characters of our lives. So the central dogma supports a concept called the primacy of DNA that says who and what we are and the fate of our lives that we lead are already pre-programmed in the DNA that we received at conception. Well, stop about this for a second and say, well, what is the consequence of this? What does this mean in our personal lives? Well, we can step back and we talk about something that science refers to as genetic determinism. This is the belief that the character and the fate of your life are already determined or described in the genes you received at fertilization. So that more or less that as the genes come together during conception, your fate is already being written in that genetic code. Well, the significance about that is when we step back and say, well, wait, the things that go on in your life, the characters and traits, you may be happy with them, you may not be happy with them, but then apparently they appear to be related to the DNA. And then I say, well, as far as you know, did you pick the DNA that you came with? And the answer is no, not really. But if the DNA provides for characters in your life that you don't like, can you change that DNA? And the answer is no, not really. And all of a sudden you start to realize something, that the character and fate of your life reflects the heredity that you were born into, the lineage, that you are actually a victim of heredity, that you yourself didn't pick the traits, but the genes are going to control them. I mean, for example, every trait that we found in the human body, there have been biologists trying to identify the genes that control them. We see that virtually every day in the papers and the media. A gene has been found to be associated with this, and a gene has been found to be associated with that. And all these traits that we're looking at appear to be written in genes. For example, scientists actually got together and looked at a group of people and scored them on the basis of happiness and tried to find out whether there was a gene that was associated with happy people that wasn't active in unhappy people. And sure enough, when they got enough happy people, they found a particular gene that seems to be more active in the happy people, and they immediately put out a big news media blip on gene for happiness discovered. And so essentially what they were trying to suggest was that the amount of happiness an individual can express in their life was genetically determined at the moment of conception. Well, all of a sudden you say, well, wait a minute. If I got a sucky happy gene, then my whole life is going to be predetermined on the basis of this heredity factor. I'm a victim of my heredity. Well, when people start to really own this, and this is exactly what we teach in school, and this is what I was also teaching in medical school, that people are controlled by these genes, then people start to realize that in some sense they're very powerless over their own life because they didn't pick the genes, they can't change the genes, and the genes apparently control who they are, so that's your fate right there. And when people start to recognize the nature of being powerless, they also start to become irresponsible, meaning, well, if I can't change it, I can't do anything about it, then who cares and why should I even bother? And all of a sudden we start to see people giving up on their lives as saying, well, it's out of my hands, it's out of my control, I have nothing to do with this. And then also making excuses for their lives by saying, well, look, I know, boss, you're calling me lazy, but I just want you to know my father was lazy. My father, he was a sloth. What can you expect from me? I mean, it's genes made me lazy. I can't do anything about it. So basically, the population has really given themselves up. And science hasn't really helped because science keeps trying to suggest that we're victims of everything. That cancer cells are victimizing us, or disease cells are victimizing us, or that bacteria can kill us, and that be afraid of this and be afraid of that because we are these, just these biochemical machines and very frail, and that nature can override this machine. And so we built into our lives the belief of victimization. I mean, even recently in Newsweek, they were talking about how fat cells are waging war on our health. And it's interesting because, well, in this epidemic of obesity, science stands back and says, well, it has nothing to do with you. It's your fat cells that are waging war in your life. So there's a tendency for us to let go of control and perceive ourselves as our fate being predetermined by the genes, and we're just trying to make the best we can out of life. And then when something goes wrong, of course, we have to go to a physician to try to get our chemistry and our genes to work more effectively to give us a better shot at life on this planet. So we feel victim to our genes, and as a result, we really express a concept of powerlessness, which is obviously very detrimental in trying to carry out your life. However, to come and save us, the Human Genome Project entered into our world. And it's interesting because even when the Human Genome Project was announced, I even thought, well, this is a humanitarian project. Yes, the idea of the project is to identify all of the genes that make up a human. And why this was going to be really important was that then if people are having negative consequences in life based on genes, then it would offer the future opportunity of genetic engineering. Once we identify all the genes that make a human, we can identify the genes for all the different traits, and that people can actually engineer themselves or engineer their offspring to create the kind of life that we would like to have, and this is actually the fulfillment of the mission statement of science, to dominate and control nature. And it's interesting because the Human Genome Project was envisioned to be the last major biology project of all time, because once all the genes were known, and we know that DNA goes to RNA, goes to protein, and that the genes as DNA would be the control devices, the rest of science would be just taking that information, learning how to turn it into technology, wherein we can genetically engineer people, plants, and animals, and create that world that we would really like to have. Well, that was actually what I thought was the basis of the Human Genome Project, a humanitarian effort. It was interesting later to find out from Paul Silverman, one of the principal architects of the Human Genome Project, that actually wasn't what the project was all about. What it was all about was simply this. It was estimated that there was going to be over 100,000 genes in the human genome, because automatically we go back to the premise that the proteins in our body, the building blocks, are made by genes. There's a different gene for each different protein building block. There were over 100,000 different proteins, so by nature there was going to be over 100,000 different genes. Plus, there were even genes that didn't make proteins but controlled the other genes. Those were called regulatory genes, and it was estimated that there were anywhere from 20 to 50,000 of those. So before the project got off the ground, the scientists were given these secure facts. Number one, there was going to be in excess of 100,000, maybe 150,000 different genes. That once we identify these specific genes, that we could then use these genes in genetic engineering experiments and correct the ills and problems that face humans in this world. Well, again, it started out as that we were going to do this for humanitarian purposes. It turns out, as Paul Silverman said, actually that wasn't the design behind the Human Genome Project. That actually the project was designed by venture capitalists for this reason. They figured that there were over 100,000 genes, that by identifying these genes and then patenting the gene sequences, that they could then sell the gene patents to the drug industry, and that the drug industry would use the genes in creating health products. So that, in fact, it was very interesting is that the program wasn't actually for advancing the human state as much as it was for making a lot of money. And that the interesting fact was that since the Human Genome Project, every gene that has been identified has since been patented as well, and that there's been an effort to try to sell these patents to drug industry for drug discovery purposes. But here's the fun part about it. The Human Genome Project got off the ground with knowing. They knew the DNA controlled life. They knew that the proteins were blueprinted by the DNA. They knew that if you go up the evolutionary scale, that simple organisms had less DNA, and when you get to the level of humans with the kind of complexity of our physiology and our behavior, that we would have a lot more DNA, so that primitive organisms would have maybe a few thousand genes, but by the time you get to the human, we're going to have 150,000 genes approximately, which in their mind meant 150,000 new drugs. So the project gets off the ground in 1987. And it actually is a very effective project, and it just really shows, again, when humans really put their heads together, they can create miracles. I mean, it was sort of like when Kennedy said we would go to the moon and made that a concerted effort. Within the decade, we end up with a rocket ship on the moon. And what's interesting about the Human Genome Project, it was one of those unfathomable projects until we decided this was going to be the effort. And then in about 14 years, we actually had the results of the human genome. And it's been a very exciting process, but it also was what I call a cosmic joke. Cosmic joke to me is when you can bet everything, including the kitchen sink, that something is going to turn right, and as soon as you make the bet, that thing turns left. So cosmic joke is when you think you actually really fully understand and know everything with such surety that all of a sudden the universe turns around and sort of gives you that little funny sense of black humor where just when you think you know it, you actually don't. Well, this happened in the Human Genome Project. It was another one of those very famous global cosmic jokes, and it goes like this. To start the Human Genome Project, at first, rather than working on humans, the idea was to work on more simple organisms so that you can get a working knowledge of how to compile the amount of information required for a Human Genome Project. Remember, the Human Genome Project is to identify the genes. The genes are the sequences of A, T, C, and G, which are the blueprints for the proteins. There are billions of A, T, C's, and G's used in the code, so it's a very large project. Billions of these bases make up the genetic code. So they started with a primitive organism. The primitive organism they use has a fancy Latin name called Caenorhabditis elegans. Caenorhabditis elegans is a miniature worm with only about 1,240 some cells that make up the entire worm. So it's a very small organism. It's barely visible with your eye. It's at the level of, it's just a small portion of a millimeter. The relevance of why they worked with the Caenorhabditis is because Caenorhabditis has been an experimental animal for geneticists because you can breed them, they reproduce very quickly and in very large numbers, and they express behavioral traits that you can study. So this makes a wonderful genetic model, and as such, because it has been a genetic model, was one of the first organisms whose genome was to be studied. When they finally finished this project, they found that this small animal that's comprised of about, as I said, over 1,200 some cells, that this animal had a genome of about 24,000 genes. And they said, well, cool, this is cool because we expect something, it's a small animal. When we get to the human, we'll have 100,000+ genes. Everything is cool. The technology of the Human Genome Project is now being established with the Caenorhabditis and appears to work. They decided to do one more genetic model, though, before doing the human, and that was the fruit fly or the Drosophila. And now most of us have done experiments in high school or college breeding fruit flies and are familiar with the fact that the fruit fly has been one of the darlings of the genetic community for almost 100 years. So, of course, it was ripe for being picked to have its genome looked at very early in the game because of the amount of information and science on the genetics and behavior of fruit flies. So they start to do the genome of the fly, and here's an interesting fact. Remember, The belief system of science is that genes control the characters, that a primitive organism will have fewer genes and more advanced, more complex organisms will have proportionally more genes making up their genome. The first organism they did was the barely visible, small little roundworm called Caenorhabditis elegans, and it had 24,000 genes. And then they complete the Drosophila genome and find out this organism, which is hundreds of times bigger and infinitely more complex than the worm, has only about 18,000 genes. And this was a big shock because they said, wait, according to our belief system, as you get into more complex organisms, you're going to have more genes. And what they found is that the primitive worm had 24,000 genes, and this flying machine called Drosophila had only 18,000 genes. And they didn't understand what that meant, but at that point, they were really so gung-ho to do the Human Genome Project, they just put that on the back burner and said, okay, we'll deal with these issues later. How can a more complex organism have fewer genes? And they start the work on the Human Genome Project. And the Human Genome Project results come in 2001, and it's a major shock. And the reason why it's a major shock is simply this, is that what they find is that in the human genome, there are only about 25,000 genes. They expected over 100,000, near 150,000 genes, and when the project was completed, there were only about 25,000 genes. And it was a big shock, such a shock to the system that people actually didn't talk about it. Well, there was a lot of hoopla. Oh, we completed the Human Genome Project, which they did. No one really talked about the fact of, what about the missing genes? There were over 100,000 missing genes, meaning if the belief system, which we really bought into about how life works, required over 100,000 genes, how can you explain a human being with only 25,000 genes? 1,000 genes more than the most primitive organism, the animal they studied, the Caenorhabditis elegans. It didn't make any sense. Matter of fact, what was so interesting about it was that there was a complete lack of discussion in the scientific journals about the absence of all these genes. It threw a monkey wrench into the system because it said there was no way then to understand the complexity of a human only on 25,000 genes, especially using the belief system that we were coming with. So it was interesting because while I had been realizing that genes were not the complete story because my cells in my tissue cultures were being more or less controlled by the environment, there was a component of the environment that has not been considered. Well, when the Genome Project came out and they realized there were not enough genes to account for human complexity, it really shook the foundation of biology, although it wasn't really publicly discussed. It was almost like an embarrassment as to, well, how can you get a human with so few genes? And so really what we started to recognize was that the genetic sky was falling. And I was out there like Chicken Little saying, oh, the genetic sky's falling. And people would look at me, yeah, but you're Chicken Little. And it was interesting because in the issue of the Human Genome Project that was published in the journal Nature, there was an article by David Baltimore. David Baltimore is a Nobel Prize-winning geneticist. So in this particular case, he's chicken big. And David Baltimore brought this one particular point up in a few short paragraphs. He said, how can we explain the characters of human biology, are things such as our consciousness and our ability to make complex changes in our physiology and account for all of our physiology and our biology with so few genes. And what David Baltimore started to realize was that we really don't have an understanding about what causes humanness to come from genes. And this was the first admission from the head of science, in a sense, or at least he being a Nobel Prize-winning geneticist, as a spokesperson for the genetics community, to say, there's something awry here. We don't understand what is responsible for human complexity. And this is such a wonderful point of view because he was the first to actually admit that while other people were playing with it. Why is this so important? Well, the interesting thing is, for me as a person who was teaching in a medical school, why this was important is simply this. Let's just stop for a second and consider a piece of logic. If a healing modality is basing its healing practice on the way that science actually works, the way that nature actually works, then by using that healing modality, it should be a benefit to the patients. In contrast, If a healing modality is based on principles that are not supported by science, then it is likely that this healing modality would actually be a detriment when applied to a patient. So it basically says if a science is based on the way life really works, that science would be good for you to be used in a medical practice, and that if you base your science on wrong information, then that science could be detrimental. And then when we consider that, then we step back and look at something that has not really been voiced very much, but has been a major concern in my life, and that is this. It is a recognized fact that conventional allopathic medicine, the medicine we use in Western civilization, is contributing to being the leading cause of death in the United States, for example. It's also responsible for 1 out of 5 deaths in Australia. That we're beginning to find that the medical profession is actually quite a scary situation because it is the leading cause of death. And the significance about that is that it was first recognized to be the 3rd leading cause of death by medicine itself. In the Journal of the American Medical Association, Dr. Barbara Starfield wrote an article revealing that from conservative estimates, the practice of medicine is responsible for the 3rd leading cause of death in the United States. It's called iatrogenic illness, meaning illness related from hospital or medical treatment, meaning a patient goes in with a problem A, but dies from problem B, something that is a result of medical treatment. So iatrogenic illness is even recognized by the Medical Association to be the 3rd leading cause of death. However, there's a more recent study by Gary Null. His last name is N-U-L-L. He has a website, GaryNull.com, where his article, Death by Medicine, is published. Gary and several other medical doctors got together and said, the work by Barbara Starfield said that the numbers she provided were based on conservative estimates, and Gary Null said, wait, rather than estimates, what if I just go make a real complete listing of all of the deaths that arise from medical treatment? When Gary did this, he found that rather than being the 3rd leading cause of death, that iatrogenic illness is the 1st leading cause of death, with over 750,000 people dying from medical treatment. And why this is relevant, of course, is that what it really reflects is a point of logic. That if medicine actually knew what it was doing, it wouldn't be that lethal. And it now stands up in the light of the Human Genome Project is that we are still teaching the belief that genes control life, and that genes are responsible for all the characters and traits, and yet the Human Genome Project now, several years ago, has revealed that there are not enough genes to do that the genes cannot control for human complexity. And so, as a result, we are perpetuating a belief which we know is scientifically invalid. Genes do not necessarily control life. Well, now I take a step back and say, okay, I'm teaching genes control life to medical students. And yet while I'm doing that, I'm growing these cells in tissue culture and identifying that the cells are responding to the environment and their fate is more or less in the hands of the environment. What I had started to realize when I was growing my cells and cultures is that when I put the cultures in an unhealthy environment, the cells in those petri dishes started to get sick, they started to die, and they just were doing very, very poorly. However, if I took the same petri dish, moved it from an unhealthy environment and put it into a healthy environment, the cells would restore their vigor and thrive and grow and develop and differentiate and do all the things they were supposed to do. And so it really revealed, just as Irv had told me in my first day of growing cells and culture, he said, when the cells aren't doing right, first look at the conditions of the environment before trying to change the cells. And I started to see that this was a truth about my cells. And what then became real interesting is when we look at ourselves as individuals, We appear to be single individuals, but we're really comprised of 50 trillion cells or so as a community. And where my work in the laboratory and where our experiences in life merge is that just think of it this way. You are a petri dish, a skin-covered petri dish with 50 trillion cells in it. If you move your petri dish in a healthy environment, they'll grow, they'll thrive, and they'll do everything wonderfully. But if you take that same petri dish and move it into an unhealthy environment, that's when it will start to manifest illness and disease, and its functions will be impaired. So basically, what I started to realize is we left out the vision of the role of the environment in the experiments that we study because we really base this on genes. And in my graduate training, I also recall experiments from over 100 years ago that actually completely denied this reality of genes. And these experiments were with the early scientists, the cell scientists, who were studying marine organisms like starfish and sea urchins. They were working with their egg cells, which were large enough to manipulate. And since the nucleus, the structure inside the cell that contains the genes, is one of the largest structures in a cell, it was the object of a lot of study. And one of the things they did in these early studies was they put a micropipette, a little tiny pipette, puncture the egg cell and suck out the nucleus with all the genes. And in these experiments, what frequently would happen is when the nucleus got pulled out, this egg cell would begin to divide. And some of these enucleated cells, geneless cells, would form embryos of up to 40 to 60 cells before they stopped developing. In other cells, they found that they could remove the nucleus with all the genes and the cells would live, some of them for 2 or more months with no genes in it. So over 100 years ago, there were already experiments that revealed if you remove the genes from the cell still expressed its behavior and adapted to its environment. And why this was important is these experiments really question the fundamental nature that we are teaching in schools and medical school that genes control life. Because basically you have to say this. If you remove the genes from a cell by removing the nucleus, a process called enucleation, and it's done quite a lot today in all kinds of experiments with genetic engineering, they take the genes out of a cell. What's important about it is this. If the cell still lives, and it doesn't have the genes in it, and it's still expressing control, then the real important question is, well, what controls the cells? Obviously not the genes, because the genes are not even in there. Well, I did the same kind of experiments myself in my clone cultures. I used different processes. For example, I'd irradiate my cells with gamma rays. These are radiation waves that destroy DNA. And so my cells in these cultures were living, but their DNA was all destroyed. And guess what? They moved around, they ate, they breathe, they digest their food, they communicate with other cells, they avoid toxins. Basically, their lives are completely unperturbed at that point, and yet they don't have any genes in them. And so all of a sudden we go back to the nature of the central dogma that life is controlled by genes and say, you can't explain the results of these experiments. As I said, most of my colleagues wanted to look at my experiments as exceptions, but the reality is there can be no exceptions to the rule. If you say genes control life and you remove the genes and control still exists, then obviously it's time to look for something else other than what the genes are. What is it that's controlling life? Well, actually, we can even ask a better question. If we thought the genes were controlling life and we take the genes and the nucleus is the organelle that contains the genes, out of the cell, and the cell still lives, the question is, well, what was the function of the nucleus then? Because we took that structure out and the cell's still going. And here's what the answer is. When you take the nucleus out, you remove all the blueprints to make the protein parts. So a cell will survive until some proteins that might be necessary to keep the cell alive, because the proteins are the mechanistic part of the cell. If I lose some of the proteins that are vital to the cell's function, and I don't have the nucleus in the cell, then that means I cannot replace those proteins because the blueprints are gone. That's when the cell will start to get sick and die. So the point about it is this. Removing the nucleus in the genes does what? It prevents the cells from replacing its parts. It also prevents the cells from reproducing itself. So all of a sudden I say, wait, all the years we've been talking about the genes and the nucleus containing the genes as the elements that control life, when in fact that's not really what the function of those organelles are. That the genes are reproduction. That their function is reproducing parts and reproducing the cell. And I joke about it in my lectures because I talk about science being a male-dominated profession, and that old joke about, of course, what do males think with. And so by doing that, the male scientist created the gonad of the cell, the nucleus, to be its brain, when in fact its function is reproduction. The genes don't know what is needed by the cell, what they should do next. There is no self-actualization in a gene. A gene is not an intelligent unit. And yet, think about all the things you've heard and read, that a gene turns on and a gene controls things, that you're walking down the street and a gene is going to turn on and give you cancer, that we talk about genes as these active, intelligent elements, when in fact, genes are blueprints, strictly blueprints. And why is this relevant? Because our terminology describing the role and action of genes is based on as if they're self-actualizing intelligent beings. A gene turns on. Well, think about it this way. As a blueprint, consider this. Consider you're in an architect's office, and you're looking over this architect's shoulder, and they're working on a blueprint, and then you happen to casually ask the architect, excuse me, is that blueprint on or off? And of course he's going to look at you with a very confused look thinking, what do you mean on or off? It's a blueprint. It has neither on nor off. What's interesting about it is DNA is a blueprint. And while we talk about a gene being turned on and being turned off, there is no on and off. A blueprint is a blueprint. If blueprints were self-actualizing, then think about it this way. That architect could take the blueprints he's drawing up, drive by a building site, throw the blueprints into the property, and come back a month later and find the building done. But the reality is, no, blueprints are not self-actualizing controlling devices. What you have is a blueprint, but then you have a contractor who reads the blueprints and assembles the building. What the Human Genome Project revealed was, the genes are the blueprints, but we didn't understand the nature of the architect. What is the architect? Who is the architect that takes the genes and turns it back into life? Well, this is where my research on cloned cells starts to come into play here, because it said that the fate of the cells, their architectural appearance, their structure, is really determined by the environment. So the question is, how does environmental information lead to the activation and control of genes? Well, this is where I started to realize that I had trouble staying in my conventional academic position, because as I started to really go into the question of looking for what controls life, and this was before the Human Genome Project came out. In fact, I left the university in 1980, 7 years before the Human Genome Project was started, because I already was aware that genes didn't control life. I was already aware that I could remove the genes from the cell and still have the control. I was aware that the environment was influential, but then my colleagues looked at me as not just being a radical, but a heretic. I was actually called a heretic. And for me, that was like a confusing moment, because I'm thinking, I'm in science. How can there be a concept of a heretic in science? And then it finally dawned on me when I read the description of the word dogma, because the fundamental belief that we were operating from, DNA goes to RNA, goes to protein, called the central dogma, is what I was teaching. When I finally decided to read the definition of dogma, I was shocked. Because the definition of dogma was a truth based on religious persuasion and not scientific fact. And all of a sudden I said, oh my God, all the years I've been teaching in medical school, I've been teaching religion, teaching the fact that genes control life, even calling it the dogma. And of course, now that I decided that from my experiments that genes weren't controlling the life, I was a heretic because I was conflicting with the dogma. And this became a whole religious argument. At some point, the religiosity of where I was led me to resign my position. And it was interesting because when I resigned my position and being outside of the confines of academic belief with its blinders to focus on what is the conventional belief of the time, I had great opportunities to expand my horizons, That's when I started to read into the physics and get information about the mechanics of life. That's when I started to advance into understanding about brain function and neuroscience, because what I was really trying to find out is, where's the brain of the cell? Because here's a simple fact. If you take an organism and remove its brain, by definition, you take that brain and throw it away, that organism is going to die, because the function of the brain is to coordinate and integrate all the activities of the living organism to keep it alive. So an organism without a brain is dead. Well, here's the point. We've been teaching, and I've been teaching personally, that the nucleus with all its genes was the brain of the cell. And yet the reality is, in my enucleation experiments, is the equivalent of taking the brain out and throwing it away. And yet when I did that, it didn't change the behavior of the cells. They still survived. They only died later when they weren't able to reproduce the parts. So basically, these kind of experiments completely deny the concept that the cell is controlled by the genes. And in fact, as we talked about, the genes were really the gonad function of the cell, not the brain of the cell. So there I am, outside of the convention, and beginning an opportunity to study, well, where is the brain of the cell? What is it that controls life? Well, I've been working with, up to this time, human cells and cells from birds and from other mammals such as mice and rats. And these cells are very complex cells in evolution, in the sense that these higher forms of animal cells are the kinds of cells with lots of things in their cytoplasm, like the nucleus we talked about, organelles like mitochondria, other organelles that have weird names like endoplasmic reticulum and Golgi. The cell is filled up with a lot of organelles. Interesting point, organelles mean diminutive organs. And so it's interesting because for every organ that we have in the human body that carries out a function, there's an organelle in the cell, meaning small organ, that carries out the same functions. Well, my pursuit is, well, which of these organelles is serving as the brain, realizing that the nucleus can be removed and the cell still is alive? I started to realize that working with these higher forms of cells was still very difficult because of the large number of different inclusions of organelles of different types that are found in the cell. I went back to a more primitive cell, to a bacterium. A bacterial cell doesn't have organelles in it. It doesn't have a nucleus. It doesn't have mitochondria. It doesn't have any real structure. Bacterial cells are just a little membrane-bound piece of cytoplasm. And the significance about it is, despite its simplicity and its structure, just an organized membrane surrounding a droplet of cytoplasmic components, these bacteria have very complex lives. They have digestive systems, respiratory systems, excretory systems. They have reproductive systems. They even have nervous systems. Bacteria read the environment and respond to the environment by changing their behavior and changing their functions. By that definition, something in that little miniature cell with hardly anything in it except a membrane and a drop of cytoplasm, something is controlling all of these functions. Somewhere there's a digestive system, a respiratory, excretory, and a brain. And what my research really revealed was simply this. Well, there was only two components to observe in the bacterium, the membrane and the cytoplasm. The cytoplasm didn't have any organelles. It was just like a solution of proteins and building blocks and energy supply and some aids for reproducing some of the parts. So all of a sudden I said, well, wait, but where's the digestive system coming from? Where's the respiratory system coming from? Where's the brain? And the only structure left was the membrane, the skin of the cell. Well, an interesting understanding in the kind of biology I was involved with, where I was studying the structure and biochemistry of organisms, is what we realize in conventional biology is that the structure of a system implies what the function is. And the function requires a structure, so structure and function are intimately tied together. And there's been a consistent understanding, the more complex a structure, the more complex the function. When it comes to the cell membrane, this is where it got very interesting, because the cell membrane is so thin a skin that you can't even see it with a regular microscope. It really requires an electron microscope to see this thing because it's only angstroms thick, and angstroms are the units of length between two carbon atoms. So if you have a chemical compound with two carbon atoms, an angstrom is the distance between two carbon atoms. And what the membrane was about 700 angstroms thick. And so it wasn't even visible with a light microscope. And when the electron microscope was developed, then it was found that every living organism on this planet, all of them have a cell membrane. In regard to the other components like mitochondria and nucleus Golgi, they're variable from one organism to another organism, but there's one uniform organelle, the cell membrane. It has the same basic structure in a bacterium as it has in a human cell. It is essentially the first organelle to evolve, and it was required for all living things. Yet, when regard to structure and function, when you looked at this organelle, it looked like a little tiny piece of plastic wrap, like saran wrap, that surrounded the cytoplasm. And so, for the longest time, biologists assumed that this little tiny, nondescript membrane was nothing more than just an envelope to contain the cytoplasm. So it represented a skin, a barrier, but apparently it must have had holes in it because food can get in and waste matter can get out of the cell. So the membrane must have some kind of way of getting things in and out. So they perceived of the membrane as this little tiny plastic wrap with holes in it. And that was all they attributed to the function of the cell membrane. And yet when I started to look at it, I started to say, wait a minute, this cell membrane, this is the structure that is doing digestion. It's the thing that's doing respiration. It's doing excretion, and it's obviously working in the neural system in these little primitive bacteria. And so my research started to focus on the cell membrane as an information processor. And I started to study the molecular nature of this membrane and how it could respond to environmental information and how it could lead to changing the function of the cell. It was interesting because I resigned my position at the University of Wisconsin and just walked out one day after realizing that I felt out of integrity teaching basic science to medical students and giving them the belief that genes control life when clearly the results from my experiments did not support that. On my departure, I had the opportunity, as I said, to be unfettered in my research, and I started to read into various other fields to try to get an integration of information about how these cells are working. It was fun because about 3 years later, I came back to Madison because my daughter was graduating from school. And while back in Madison, I thought, well, geez, maybe I could go back and talk to my old colleagues about this new understanding I was creating about the nature of the cell membrane and how the membrane is really what processes information and controls life. And so I asked my friend who was chairman at the time, I said, listen, I'm back in town. How about letting me give a lunchtime seminar about my new story called The Membrane as an Information Processor? Well, he said, sure, come on in and give this lecture. Wednesday lectures were conventionally just a social meeting where a lot of the faculty and the students would come together on Wednesday, bring their lunch, hang out, and then somebody would give a talk, and people would pay attention or not, whatever, what was going on. It was, again, just more of a social thing, and a person could give a talk and stutter and drool, and at the end everybody would applaud and say, oh, that was wonderful. And so I figure, okay, I got an opportunity to do a Wednesday lunchtime lecture, so I show up. And I see all of my former colleagues and a lot of the students that I didn't know were there and other people I knew. And I start to give this lecture on the information processor or the membrane and talking about how genes really didn't control life. And as I was giving this lecture, about like midway through the lecture, all of a sudden it dawned on me as I was talking, I realized nobody was eating anything. They were just sitting there staring at me with like big saucer eyes. And finally I get to the conclusion and I say, thank you very much for listening. And as soon as I say that, I pause and there was dead silence. Not a person made a sound. They were all just sitting there frozen in their seats. Nobody had eaten lunch. Nobody had talked to each other. And that was that hardest silence I ever heard in my life. I was just waiting for something to happen. And all of a sudden, one person way in the back of the room started to clap. And as soon as that person clapped, everybody stared at that person. He stopped clapping. And then the next thing I realized, they all got up like it was a ceremony and they walked out on me. Every one of my colleagues walked out on me, and I felt so like, oh my God, am I that weird? Am I that crazy? It's sort of like when a soldier has done something wrong and they take him out of the army, they take his epaulets off and strip him of his buttons from his uniform and, force him to walk away. And here I was being invalidated, my lab coat buttons essentially being stripped from me, saying, you're a heretic, this is unbelievable stuff, and sent me on my way. I was completely devastated by this because I thought this was just great science about how the membrane is really controlling life. So the interesting thing is I said, okay, I might be crazy, but I started looking at the literature, and the more I looked at the literature, everything kept revealing the same thing, is that I was on this right track. So I continued this research, and I'm working on my own, and now I actually end up with a job. I'm teaching in the Caribbean on an island, Grenada, one of what we call offshore medical schools, where students that can't get into conventional schools, medical schools in the United States, have an opportunity. There are several schools that are found outside the United States where students can go in and do the work and then come back in and take the required board exams in the United States. And I was teaching at one of these schools. And while I was teaching there, I had continued my research on understanding the nature of the cell membrane. And it was on a particular night in 1985 that it was about 2 a.m. in the morning, and it's now about 10 years of research on trying to understand the nature of the cell membrane and how this structure is involved with controlling the cell. I've looked at it upside down, left and right, backwards, forwards, revealed its structure and through all the kinds of molecular analyses, studied all the movements, and I had an understanding of it, but it really didn't gel until this particular night in 1985. And in 1985, I rewrote the definition of the cell membrane as I'd been looking at it in terms of biochemistry. I had rewritten the definition in a way that I had never written it before. And I wrote down this following definition of the cell membrane. The cell membrane is a liquid crystal semiconductor with gates and channels. And I heard myself say that, and as I was typing it on the computer, it really dawned on me and said, wow, that's a real interesting definition. It's funny because I've heard that definition somewhere, but not in terms of biology. And then I started getting somewhere, I just read that. And I looked around, and there on my desk, on the corner of my desk, was my first Macintosh with this little smiley face and a book right next to it called Understanding Your Microprocessor. And this was a booklet that I bought from Radio Shack describing in simple terms how my computer worked, because that was my first computer. And all of a sudden I grabbed that book and thumbed through it, and there it was on page 3 in the introduction. There was a definition of a computer chip which read, a computer chip is a crystal semiconductor with gates and channels. And I stopped for a second. I said, oh my God, wow, what a coincidence. The definition of the cell membrane and the definition of the computer chip are exactly the same. And then all of a sudden, I started to realize that the data in my head started to spin around like crazy. And it was interesting because I got this visual image of like a slot machine at Las Vegas where you pull the crank and then all those little wheels start spinning, and then all of a sudden a cherry shows up and another cherry shows up and another, and all of a sudden all these cherries came across the screen of my mind, and it was like this jackpot, because what all of a sudden I understand was it was not a coincidence that the cell membrane is structurally and functionally homologous, meaning the same as a computer chip, that its function is exactly the same as a silicon-based computer, but this is a carbon-based computer. I started to realize that the cell was a chip and that the nucleus was a hard disk with programs. The genes were programs. But what was really exciting, as I was typing this on my computer, I realized at that particular moment, my computer was like a cell. It had programs built into it. But what was expressed by the computer wasn't determined by the programs. It was determined by the information that I, as the environment, was typing onto the keyboard. And all of a sudden, all the pieces fell into place. And I realized, oh my God, The cell membrane is actually an information processing computer chip. The cell's genes are the hard drive with all the potentials, and that's why every cell, such as a stem cell in your body, can form any kind of cell, because every nucleus has all the genes that make up a human. But why should a cell be skin, and one cell be bone, and one cell be eye? And the answer is not because of the gene programs, but because of the feedback of the information from the environment. And all of a sudden, the bigger thing hit me, and the bigger thing was this: that what makes us different from each other is the character of the keyboard on the surface of our cells. That, as we're going to get into later on and describe how this works, that the keyboard has a pin key, a pin that makes each one of us different. And the significance about it is that the pin key, the identity keys on the surface of our cells, respond to environmental information. And all of a sudden, the biggest aha that I could ever imagine hit me at that moment. And that was this: that my identity is actually an environmental signal that is playing through the keyboard on the surface of my cell and engaging my genetic programs. That I myself was not actually even inside the cell. That you are not inside your cell. You're playing through your cell using the keyboard as an interface. That you are an identity from the environment. I was an identity from the environment. Well, what was really profound about this was that when I went back in my history as to why I became a biologist, I became a biologist because in my youth and the dysfunctional developmental experiences that I had in my younger days, I realized that when I wanted to get some kind of truth, that I didn't see that religion was offering that. Because while they spoke all the words and they all sounded really nice, their behaviors in no way seemed to match what they were talking about. That led me to science, and as a youth I really saw that science was important, because a truth in science is the same in New York as it is in Brazil, as it is in Germany. That all sciences are true, and once we establish facts, they become universal, and that they become accepted by all as this is a truth, rather than what I saw religion as separate, each giving different truths and not being coordinated. So I went away from spirit, which I didn't believe in, and ended up in science, only to end up in that particular night in 1985 identifying the fact that my identity was something from the environment playing through my cells. And it was the greatest shock in my world, because I was completely thrown from a non-spiritual reality into the requirement of a spiritual reality based on the fact that I was not inside the cell, I was playing through the cell, meaning this: my cell was like a little television set with antennas, and that I was a broadcast, and that my broadcast station was picked up by the antennas, and that controlled the readout of the genes, so that I was actually programming my cells. What was relevant was I realized that if the cell died, it didn't necessarily mean the loss of the broadcast, that the broadcast is out there whether the cell was here or not. And all of a sudden this hit me with such profound awe that I actually just had this complete emotional transformation, because at 1:59 in the morning, here I was, non-spiritual, looking at the mechanistic side of life, and at 2 a.m. in the morning, all of a sudden realizing the requirement and existence of an identity that was outside of the cell and independent of the cell. And I was just completely blew my mind. I was so out of it, I couldn't believe it. I mean, I started to realize, for example, the first important thing was a weight was lifted off my shoulder, a weight of mortality. I didn't realize it, none of us really realize it, that our mortality is very principal element in guiding everything we do, because every step you take, your biology is reflecting on whether this step is going to enhance your survival or compromise your survival. What I realized was when survival wasn't that important, because I realized my eternal character is part of some broadcast in the field, this weight, this fear of mortality disappeared. And it's been great, because that's been about 25 years ago, and it's just one of the most wonderful, liberating experiences I ever had, because it took away the nature of mortality. It added more to who I was and more value to what my life was. But this was just so exciting. I really wanted to tell somebody about it. I was living on this piece of beach way off on the edge of Grenada. I didn't have a phone. It was 2 o'clock in the morning and wasn't anybody I was going to call anyway. And I thought, who could I tell? There wasn't anybody there. I just got so excited. And I remember, well, it's a medical school, where I'm teaching, that students are up all night. So I got in my car and I drove off to the medical school because I had to tell somebody about this exciting discovery, about this new understanding about how the membrane controls life and the spiritual character. And I drive up to the school, and sure enough, the lights are on the library, and I run in the library. I'm sure my hair was all up. My eyes were wide open. I was like, oh, so excited, this great secret of life. And I grabbed the first-year student. I said, listen to this. And I started explaining about the nature of the cell membrane and the crystal structure and all the idea about the physics and the Fourier transforms and things that he didn't understand a word I was talking about. And he looked up at me after I took a break, and he said, are you okay, Dr. Lipton? He thought I was so wigged out that I might need medical attention at this point. And yet it was so amazing to come across this piece of information and have no one to communicate with. So started off on this whole preamble to get to this point of actually telling you a new biology. And this new biology deals with the fact that the brain of the cell is the skin of the cell. The skin of the cell is called the membrane. And it's interesting because this skin is what separates the inside of the cell from the outside world. And in this particular position, the skin becomes very important because the skin is the interface of two dynamic realms, the interior of the body, the self, and the exterior ever-changing world that we live in. And as in its position, the skin can see both environments at the same time, read the conditions of each environment, and then serve as a device to read what's going on in the outside world and adjust what's going on in the inside world to keep us alive. So the skin of the cell is the brain of the cell, and that is the functional element that controls life. And why this is relevant is because it's a process of dynamic control as compared to the concept that we were dealing with before with genes, where there was a program established at conception and the rest of our lives was just essentially a readout of that program. The membrane, as controlling of genes, offered new insights. The membrane could select genes and deselect genes. The membrane could control genes. So basically, what we're going to talk about from this point on is the nature of how this membrane or brain of the cell works. And the reason why this is important is because when you understand the nature of it, you'll start to realize we are not victims of our genes. That through the action of the membrane, we can actually control our genes and our biology and our life. And we have been doing it all the time, although we've been laboring under the belief that we are victims. And why this science becomes really interesting is because it reveals how the membrane perceives the environmental signals and then translates those signals into biological signals that control our biology. And so that where this really becomes relevant to us is that we perceive the environment and adjust our biology. And as we'll get into a little bit later, we'll recognize a very important fact. Not all of our perceptions are accurate. That we acquire perceptions that are actually misperceptions. The relevance about that is, since the biology converts perceptions into biology, if we are laboring under misperceptions, then those misperceptions adjust our biology, but actually provide for a misadjustment of our biology. So when our perceptions are inaccurate, we can actually destroy our own biology. Then when we turn around and recognize, since perceptions control life, then think about it this way. The old vision of genes controlling life, to change life, we have to change the genes, the Human Genome Project, genetic engineering. But when we understand that genes are just respondents to the environment from the perceptions handled by the membrane, then we can realize that if life isn't going well, then what we really have to do is not change our genes, but change our perceptions. And that's a heck of a lot easier to do than physically messing with the body. And in fact, this is the power of this new biology, that we can control our lives by controlling our perceptions.
This concludes Session 1 of The Wisdom of Your Cells. Our program continues with Session 2, The Assumptions of the Apocalypse.